Thanks so much, Heba, for the kind invitation and introduction. Really looking forward to some discussion here on all these spatial omics developments and potential avenues. And before I start, maybe sort of just as a key point, I think with this whole multimodal spatial imaging field, we're sort of at a state that single-cell genomics or particular transcriptomics has been something like 5 years ago, where there's sort of a bunch of competing techniques and there's no real standard yet. And in particular, like microscopy, there's like so many standards, and then we don't really know how to relate to that. Like at the time was bulk and single-cell RNA-seq and so on. And I think some tools sort of need to be developed. So what I want to show today is one of our approaches, how we think we can sort of organize, at least within the lab and hopefully also with a few partners, how we deal with these type of things. Let me just like share my screen here. I hope you can see the slide.
Yes, perfect.
Thank you so much. So I want to talk a bit more generally, not only about this tool that we've been recently doing, which is called Squidpy, but it's sort of a bit more generally also about what we've been, sort of how we're thinking about these type of spatial data sets and what we can be doing with this. And I entitled this learning the spatial components of molecular tissue biology. This sort of is a reminiscence of a sort of simpler, sort of the simpler world of disassociated RNA-seq, where there was also a paper entitled like this coming out. So let me get started. You know, the whole single-cell biology by itself obviously started from the microscope when we saw the first cells. And this beautiful review from Alex van Oudenaarde, every cell is special. He's been sort of pointing out that, you know, these two worlds, the omics world and the single cell world, which were very far apart over time. And, you know, this is like 6 or 8 years ago now, has been converging towards these many genes, many single cells with single cell genomics, but then also been followed up with the whole spatial transcriptomics and now multi-omics and so on starting to happen. So I think it's very nice where these go together. And, you know, also, of course, in genomics, at the heart of it, there's also a microscope. So, you know, it's quite nice how these things actually come together. And I think studying cells in their natural context, but in a potentially more unbiased fashion is very exciting. So no wonder this has been named one of the method of the year just like 2 years ago. And I think Nature just highlighted one of the upcoming big things will be spatial multi-omics, what people start now doing. So, you know, just like add on top of it, as biology and biotechnology often does, it of course gives rise to fun analysis challenges. My lab has been doing a bunch of things, but one that has been actually having an astonishing nice impact is this tool called ScanPy, which we actually set up to study, sort of to support analysis workflows in the single-cell RNA-seq and genomic setting. And this has been really nice and useful for organizing stuff within the lab, but has been also picked up quite nicely. And so we'll be building upon this. So this was this review at the time from Nir Yosef and also Aviva Gelb's lab that was actually getting some attention. And this was sort of nicely saying that, you know, in single cell genomics, you want to study different areas of variation. They sort of mentioned pattern already, but I think based on this, we've been trying to think what would be this equivalent in the spatial setting. You know, in the single cell RNA-seq setting, very often we talk about cell types and the cell clusters or so as one of the common ways we talk about coarse grain interpreting these data sets. I think one interesting aspect before I now speak about the more general setting is that in this thing here, what we actually notice, because these techniques become so common, we actually get to do this across many labs. And if I was to summarize a big advances in computational single-cell genomics, one of the big things was that people have been thinking how to deal with these multiple batch data sets. So for example, here, you know, if we look in a single setting, we have a bunch of cell types within one lab. If we do this now across many studies or labs, suddenly stuff is all over and there's like a strong batch effect driving a lot of the variation. And we want to remove this variation. We're not caring about the technical variation very often. So this is like, has been termed data integration. And a bunch of methods have been coming up for that. And we've been, for example, also benchmarking these type of methods. Without now saying what we've been doing in detail, just mentioning that a lot of work from particular Malte, a very talented postdoc in the lab who's about to set up now his own lab, we've been trying to really think how we can integrate data sets and sort of think about different ways. We set up a competition at NeurIP's large machine learning conference, or one of the biggest machine learning conferences, where we had a multi-omics single cell data set, both RNA attack as well as RNA protein, and asked about challenges to the community. How can we cross-modality prediction, matching cells, or doing joint basis-based learning? And the interesting thing is that all of these winning methods, which is briefly depicted here, all of them were using in one way or another a neural network for doing so. So if I understand correctly, many of you might be coming from the so from the spatial side anyway, then it's no wonder that in computer vision, neural networks are the thing to do a lot of analysis. In our field of the sort of disassociated setting, this has been only somewhat more slowly picking up. And maybe let me say a little bit towards that before I then speak about the concrete tools. So for that, I think, in a sense, unsupervised machine learning, this is sort of the task that we need to do here, is something like variation learning. So just finding a latent space in these single cell RNA-seq data sets. And this is the common architecture that particularly near Joseph, as well as my lab have been sort of putting forward early on, namely taking a gene by cell matrix, then sort of squeezing it down, this bottleneck, blowing it up again, and making this as similar as possible to the input. If you do that, for example, you can reconstruct or clean up cells. You can do multiple additional things. And the interesting part is that in this bottleneck layer, there actually you remove variation. So you sort of understand the core variation in the data set. So let's start this latent space or this core variation learning. For example, in this process, it's sort of very sparsely depicted here. You might be finding in this differentiation program. Or in this data integration thing that I was mentioning before, this thing might maybe move some of these varied data sets together and really integrate them better. And we've been building all kinds of things on top of it, such as studying variation. And I won't go into detail now for this thing, like we've been studying perturbations and so on top of it. It's really fun. But what I wanted to say is that these tools are now around and actually can help us also already in this disassociated setting to interpret things. And the question is now, how can we use these type of methods also in the spatial setting? So in this review of spatial components of molecular tissue biology, that was our idea of following up this older review together with Aviv Regev. It was just coming out a few weeks ago. We've been trying to now say, hey, if we now have these spatial observations, what additional axes of variations can we do, and how can we model this? So how can we set up sort of a model that would maybe take spatial information as well as this covariant information to then predict or model one particular readout, which could be gene expression or maybe some subcellular localization of gene expression, maybe also morphology or also sample variation, and then just say communication of more complex dependencies. I think this type of formulation would really help us to, let's say, correlate in a simple setting, or maybe also then do some more complex predictions and sort of models of all of these axes of variations. So we understand how variation across space then impacts these type of observations. And if we do this, maybe we can ask sort of very localized questions, such as, which cells would be interacting in a local pattern, or would we sort of maybe have more long-reaching effects, or also which type of sort of similar enrichments of cell types would maybe form in particular tissue things. And here you see already that scale is going to be a big question. And at the recent CCI meeting, actually, a bunch of sort of people from the community came together and tried to define questions. We've been really asking, what is the best notion of a cellular niche? These things sort of try to look for niches, but what's the scale? Like if something diffuses, it could be much larger than maybe a simple thing. Or, also how do we find these building blocks? With this associated setting was very simple. We just look for clusters, roughly. But even that sort of cost the community quite some time. What's the equivalent here? And with Squidpy and sort of some downstream tools, we've been trying to put some spatial statistics and so on in the hands of the users, but I'm not sure we really are able to robustly answer this yet. In any case, if we now have these type of axis variation, we can ask things. We can sort of, for example, see how does gene expression, let's say localized in one cell, vary across spatial dimension, maybe on the short or on the sort of more longer term. And you can look, for example, for variation of some type of morphogenetic factors, activity of T cells or some type of toxicity and so on. We can see how things change in a tumor environment. Similarly, what we have been doing for a long time is arranging cells by a similarity in terms of expression profile. That was helping us to find, let's say, here progenitor cells that would be differentiating to downstream populations by maybe some sort of limited cell type. I guess you could also ask, how can we do the same thing in space? Could we, even though we just see a snapshot, maybe learn that this cell was then maybe sort of originating, it's like a stem cell, was always proliferating and then pushing cells over there that would sort of then migrate to these things? So could we also have methods to reconstruct that? Similarly, and this was then sort of our summary of all the different analytical challenges or maybe also opportunities that could be posed in this. Clearly, because in contrast to the disassociated setting, first we really with any of these tools, need to invest in this image processing, segmentation, registration, maybe also on a common coordinate framework. These type of things, I think, is an important first step for analysis. As the next one, depending on the spatial assay that you'd be doing, you might be doing some type of more deconvolution, or also this integration that I was speaking about, sort of adding multiple data sets, or sort of seeing you in one spot where there might be multiple cells overlapping, you sort of put the different cell type frequencies in, obviously with subcellular imaging techniques such as Murphy's, so you might not need to have to do this. You might want to do multimodal analysis because you have different observations included. And then you might be asking, what type of features are actually changing across space, in what manner? So it's something maybe localized in one or the other region. So some very homogeneous, or sort of, just one region activated variability, and maybe those features are also correlated. You could be asking in a tissue context, what type of cells would be occurring next to each other and influence their mutual profiles. And this one, you could potentially put them also in a cell communication graph. So you just like put these into a graph and try to see, what type of expression profiles are correlated with a bit more complex. I'll be saying something about that in a minute. And of course, as we do in single cell RNA-seq, you might want to do also spatial power analysis and then design these things, because I think in the future we'll be doing much more localized things. So in the next something like 15 to 20 minutes, I want to speak about some of these examples. So I want to speak briefly about something that we do very early on, in this case, actually selecting probes for targeted assays, about our sort of general analysis framework that covers a whole bunch of these things. And if there's time, briefly about how we define actually cellular neighborhoods. I'm very excited to get your feedback on each of these things. So the first one, this is work by Louis, PhD student that works together with Ali Ertrik's lab and some postdoc Malte. And we've been asking within our DiscoVer project, so a big project where we try to build the human lung cell atlas. We've been asking, how can we find for targeted spatial transcriptomics, where you actually have an assay that does not do genome-wide for sequencing, but only looks in a bunch of probes. How can we design this based on single cell RNA-seq where we know already which probes are separate cell types? So we want to find the best set of features that is actually doing a good job for sort of differentiating between those clusters, but then maybe also have still sufficient variability and sort of interestingness and find additional information. So for this, we've been coming up with, there's a bunch of ideas what you could be doing. What we came up with was a selection procedure where we start actually with the set of genes, and then actually could check for main variability, just as you see before, the components of variation. In one sense, we look for differentially expressed ones across the clusters and integrate them with the tree and add them if needed into also just a base loading to really expand the set of genes that have most of the variation there. And we would be adding additional information to this process, to this probe set, because of maybe literally defined genes or sort of probes that are interested for user. And this type of thing we then compare to simpler type of approaches based on all kinds of evaluation metrics. For example, how good we can recover variability, how good we can identify cell types, and also how long this thing will take. So we've been setting this up on a data set. And again, I'll be short that this is all unpublished, but I think it might be useful thing. We've been setting this up on a human lung data set together with the Kerstin Meyer and Tangram lab. So we have a reference set, a set of cells of 8,000 genes and a design set of probe sets just down to 150. So very strong reduction. And you kind of see that the clusters, obviously, these humans look different because there's some indeterminacy there, but you kind of seem to reconstruct pretty much the same structures. And it's visual, so it's maybe not the best thing to do. So of course you can quantify this. In this case, we've been just doing this for our probe selection method called SPARPROS. That's the current name. We might be discussing this a bit more. Selecting for 50 genes and then comparing to the simpler things such as differential expression, a forest-based setting, PCA, or maybe some other type of things, how you would be doing feature selection. And in terms of both how good we get the overall variability, but also how good we pick up the particular cell types. There's multiple stats that you can do for this. But what's nice to see is that actually this type of approach that we're doing in many cases actually picks up very nice both the set of cell types as well as variability. And if you sort of aggregate this by some type of score, this is actually also consistent, even those sort of underlying things then would match across the number of genes that you would be selecting. Of course, it's hard to really define proper ground truth, so that's why we sort of get these metrics in here. But we can then apply this for a real spatial experiment. In this case, a screenshot selection that has been proposed by the Samarkov's lab, and that we actually identify a bunch of factors here that you would be just defining in red from the single cell setting and from the spatial setting. And you see that in many cases, those profiles will identify the same cell types and still capture sufficient variability. So that's something for probe selection. If you want to learn more or sort of uses that we know, and let me now come to this more software type of thing. So once we've been observed and generating our spatial spatially resolved single cell data, we want to do some analysis. And ideally, you don't want to be sort of tied down to one particular setting that's maybe vendor specific, but you want to have a data structure that's compatible, maybe that you can sort of plug in extending tools, and you sort of want to embed this in some type of tool chain. And we have been for a long time believers in actually doing this robustly for the single cell RNA-seq setting. That's why we set up the single cell analysis in Python software. Now, if we add space, we've been discussing the group what would be a good name for this, and because I have very funny PhD students, they were thinking this ScanPy sounds like Scampy, you know, this animal here. That's the logo for this thing. So let's come up with some other fish and we call it Squidpy. This is work by Giovanni and Hannah, but essentially the whole team. We're doing a hackathon where we've been developing this set of tools and they've been carrying it forward. It just came out this year. The idea there is that we take a whole bunch of spatial omics data, which is rather methods independent and can read all kind of things. And if your technique is not there, contact us. We're happy to provide a loader for that. But essentially then generate either these sort of very regular or some more complex segmented data sets. And it really has a whole bunch of interfaces to popular pre-processing tools that do, for example, very much focus on segmentation and processing, such as Space Ranger for Visium, but then also Starfish, Napari, MC tools, and so on. And once we have it in there, we have a storage format that builds upon our ScanPy and data object, but also has a spatial graph and an image container that captures much richer information that you have in images than just the single cell expression profiles where we have aggregate things. So, you know, we have as a unit of measurement, you have the cells in terms of the whole profile, but you also annotate local spatial neighborhood and you can do something like that. And current methods, I believe, in terms of analysis, mostly now revolve around this image processing, segmentation, registration, and then deconvolution and integration, and then this spatial variable feature analysis. And this particularly this thing, you know, how to add space in terms of analysis, we've been trying to realize by then the set of tools that are enabled by Squidpy, which is sort of all building upon this infrastructure, and it's modular and extensible. So, you know, people can build upon this data structure, and there's a bunch of things that are already starting to taking stuff. But one of the most obvious ones would be obviously just want to visualize that. And for this, we plug into, you can do all kinds of things, but the thing we recommend is plugging into Napari and image viewer developed by CCI, which is that gives you all kinds of flexible ways of visualizing this multimodal data. And then you can local spatial statistics. You can do sort of just see, hey, you know, which gene is enriched where, or something like that. You can maybe look for close by cells that have the enrichment with respect to particular ligand receptors, thereby sort of learning something about cell interaction and cell communication now in a direct fashion, much more direct than you would maybe have in this disassociated setting. But then you can also take image features into account and model some of these. And because this is done in Python and sort of been written up properly, you can very easily interface also with more complex machine learning systems and maybe do your classifier on top of that or something. So this is this framework for spatial cell mapping. As I said before, we have all kinds of architects. So we have all kinds of ways how to formulate this image container, which goes beyond just the AnnData. Could have different type of grids, also generic architectures, how to store them, these things. We have a pipeline from pre-processing, segmentation, and feature extraction to then also this visualization. And just to show you what you can be doing, just a few highlights. For example, you can ask for spatially variable genes. So just pick me those genes that are really doing something spatially interesting, not just like salt and pepper. For this, you can, for example, take more in spatial autocorrelation. You can see to pick up genes interesting thing. In this case here, this is a mouse cortex data, and you can identify some that are actually doing have this sort of interesting patterns here in the Visium data set. We can look for ligand receptor reactions. For this, we implemented this original disassociated setting paper from Python lab called CellPhoneDB, but made it sort of more larger scale in terms of the annotated ligand receptors that come in by building upon OmniPath resource. But it also came up with sort of fast implementation and can quickly now scale this across all regions. We can analyze really large tissues and do segmentation feature extraction. Been implementing something very simple there with respect to pre-processing and segmentation, just watershed and a deep learning based one. But of course, I think segmentation is like a whole subfield by itself, and there's all kinds of things out there in this module, and you can plug your other things in, and then you sort of get a nice summary of those results. You might be interested at actually doing something also with the image readout. So not only the multimodal spatial profile for the for one cell. So what you can do, you cannot take those image features and put it also in a scan pipeline, and then say, you know, I don't want to just make clusters based on the observed gene expressions for each dot, which would be then sort of giving me this, but I want to have similarities of local image patches. Let's say capturing something like cell morphometry locally, something like that, and then you can cluster things, and then those would be potentially more spatial coherent, but there might be sort of also finding some interesting things beyond that. And you can also take bigger sort of pre-trained networks such as ResNet and embed those clusters, so you can find sort of similar shapes locally. You can do all of this sort of computation nicely. I think I skip over that, but I think there's a bunch of applications that could be directly useful. For example, really, if you want to try this out on these sort of spot-based assays, such as a recent data set, you might want to deconvolve things, and for this you have an interface with Tangram and as well as Celltype location. You're just showing the Tangram one, where you would be looking at coordinate spatial coordinates, build this reference atlas, and then you deconvolve it, and then you get sort of for each of your particular deconvolved cell type proportions this local visualization. You can look at this deconvolved segmentation. You see that you sort of see a layering thing with respect to the different cell types. I think an important point is obviously just to make sense out of it. I think this meeting is called BioVis, right? So visualization, I think, something you might be particularly caring about. And in this case, you just like plug this thing in Napari, you load it directly from your Jupyter notebook or something. And then you can highlight particular genes where they would be localized. For example, again, here in the vision data set, you can change layer settings, and you can sort of look at the underlying H&E stain. And you could then potentially also look at annotated information, for example, as we store the AnnData also cluster annotation and so on. You can also do annotation within this using Napari. So for example, you could try to see where's an interesting gene here, and you want to then maybe pick this region of interest. And for this, you know, Napari has a bunch of tools that you can directly do, and Duani and Hannes and I see sort of made an interface to then export this and then have this sort of ROI also in your AnnData and then downstream analyze it. There's a bunch of documentation, so this thing has been set up properly. I don't think I have to say this, but I think it potentially so. But obviously, there's all kinds of limitations still, and I just want to spend two minutes on this as well. For example, what we currently still do is that we store and analyze at the moment representations aggregated per cell. It might be actually more interesting to have molecule level representation. So really the subcellular information that you might get from a lot of fish-based data, and some people are interested in that. For this, you need to sort of a hierarchical type of storage system. And for this, we're thinking about extending the AnnData using something called orbit arrays. I think large microscopy images are kind of doing via this image container. I think there could be more clever ways to do so. And so for this, we're thinking about actually changing this this type of internal architecture. This Napari plugin would potentially need an extension towards more larger scale microscopy images, and we're thinking about also registration module for multi-slide data sets. We're currently working on each of these things, and just sort of to highlight a few of those. You know, this AnnData object is super popular for this sort of disassociated settings. We're thinking about sort of putting this into this OME-Next Generation File Format for imaging, to really then make this sort of compatible with the spatial objects and sort of bring these things together here, and then also make this sort of available in a bunch of viewers. Yeah, and I think I can skip this connection to other ecosystems. I think I mentioned this before. So in the remaining 5 minutes, if that's okay, I just want to point out one more thing, what you can be doing with this. So this is enabling a lot of things, but this, of course, still very much of the infrastructure. I think one interesting point, that's why I highlighted the whole deep learning things in the beginning, is that those models are very flexible. And there's been a paper recently from Al-Quraishi, one of the developers of this Rosetta prediction of protein folding, as well as Peter Sorger, outstanding colleague in systems biology, systems medicine. They've been putting this idea forward of what they call differential biology, that you can actually employ these deep learning models not only in sort of very other settings for molecular sort of spatial structural bioinformatics, but also in our setting. They've been thinking you could potentially combine this with mechanism. That's a bit of an oxymoron, right? So you have here the sort of super flexible black box type of model, here the mechanistic ones. How can you bring things together? We've been all thinking about some type of constraints on this. And what they've been saying is, I think it's a nice point, is that like with all these sort of things that you maybe have convolutional in the imaging setting, or group equivariant in the spatial setting, or recurrent text setting or sequence setting, we've been adding these constraints anyway. And maybe more generally, we have a lot of these systems, old school principle-based models and these data-driven ones. Maybe that's a way to connect it along these similar type of constraints, where you maybe then, that's what they call differential programming, concatenate a few of these ideas and constrain the models with respect to both of these aspects. And I like that a lot. And I just want to sort of show you one thing where we've been trying to do a little bit like something like that. So you want to sort of add constraints to it. And one way of adding constraints is that we have this spatial graph structure, right? So we put the spatial structure essentially to a graph. So maybe what we can do, and this is the idea of David and Anna in this particular setting, that we want to take some spatially labeled system, either single cell resolved, so something such as a fish-based assay. We can do it also for Visium ones after deconvolution. Won't be showing that. But in any case, ask them, is the cell actually interacting with this one? How can you predict this type of interaction if you really observe each of these sort of activities as the expression will be? So for this, we build a spatial graph of cell types. And then we ask, can we use this spatial cell type graph to predict the gene expression at one particular point if we take all these neighbors into account? So we want to write up a function that predicts based on cell type by itself, as well as the niche, the gene expression of the thing. And if you was just not allowed to use the niche, here would be just like predict from the mean the actual expressions, it would be just sort of a central prediction. But if it takes these into account, it could be sort of a bit more specific. And if you then find something where this is actually possible, you know that there's some interaction happening. So we want to sort of decorrelate the intrinsic variation from the extrinsic variation. We will be doing this on this type of image. So you will be learning spatial representation using these graph neural networks. Here we have a meetup data set from Hartmann et al. I think in colorectal carcinoma, where we first see labeled cell types. We then project them just on their spatial coordinates and build this local, in this case, irregular cell-cell graph where we can then ask for these communications. Similarly, here on a MEFISH data set for mouse brain, where you see sort of all these nice layers, then you can build this communication graph and ask where cells will be localized. And what you can now do, and this is sort of the last slide, I really don't want to go more. What you said here is you can now ask how much of intra-cell type variation can you explain? So how much can this functional prediction actually tell you if you add using external sort of neighboring cell type expressions? So in this case, we ran 3 different models, and here only allowed the disassociated things and no spatial information. You'd be getting that. And here we'd be adding also information about the scale of 10 microns, 100 microns, and so on. And what you see is if you go a little bit beyond the sort of typical cell size in the region of 100 microns, so these things actually has most of the information and goes down again, which tells you that it seems to be picking up local order of gene interactions. And what's nice, it's actually consistent across a whole bunch of different measurement techniques, and it always peaks at sort of an interesting scale. And then you can learn something about interactions and so on. Which particular cells are doing this, which I will skip now. And just to conclude, so I've been starting by mentioning computational challenge, which aim to find components of spatial variation. Spoke a bit about probe set selection as one of the tools for doing efficient spatial transcriptomics. Squidpy to enable actually some type of assay agnostic spatial analysis, and spoke about how do we use graph convolutions to have neural networks also in the spatial setting. I think where things will be going, of course, to make those agent space more interpretable, and then also studying maybe tissue structure or some higher level variation. This is my lab. This is how Bavaria, where we are localized, mostly looks at this sunshine. Happy to discuss further later. Thanks for the invitation.